Good afternoon, everyone, and a warm welcome to this webinar. We are MedGenome, the leaders in genetic testing in India. And today's webinar is in association with Federation of Obstetricians and Gynecological Society of India, FOGSI. And this is a three-part series. This is the third webinar in a three-part series. And today's webinar is on screening for genetic disorders. And before we start, I wanted to inform the audience that if you have any questions or comments, please type them in the Q&A window of the Zoom conferencing browser, not the chat window. Q&A window, our speakers will address the questions as and when required. And all attendees will be awarded one ICOG point for this webinar. And we will be sending out across a recording of this webinar in a week or so. So please look out for it in your inbox. So now, without any further delay, I will now hand over to our chairperson, Dr. Mandakini Pradhan, who will be taking this session forward. Dr. Mandakini is the professor and head of Department of Maternal and Reproductive Health at SGPGIMS, Lucknow, Uttar Pradesh. She is the chairperson of the Genetics and Fetal Medicine Committee of FOGSI, and we are doing this webinar thanks to her initiative and vision. And she is a leading clinician in the field of genetics, and she has over 82 papers published. She has been quoted in Williams' textbook. She has been awarded multiple awards, including Bangladesh Ultrasound Society 2019, awarded Nari Shakti Award in 2020, Bharat Gaurav Samman Award in 2020, Health Icon Award from Times of India in 2018, and many, many more awards to her credit. And she aims to start a super specialization in maternal and fetal medicine. So over to you, ma'am. Please take it forward.
Yeah, very good afternoon, everyone. This is at the outset, I feel sorry about not being getting connected in time and then somehow technical difficulties. But indeed, I have got this pleasure and opportunity to introduce to call upon our guest of honor, that is Dr. Hema Divakar. And can I have the slide of Dr. Hema Divakar, please?
Yes, ma'am, it's there on the screen.
And every one of us probably know her. She is our past president, FOGSI. And even whatever she is, she shows in her work. So she works for the woman. And to this extent that menstrual hygiene, anemia correction are her pet subject. And Manyata, in fact, she is busy in opening up this Manyata project in somewhere else. So she will be going and she will be addressing. We are very fortunate to have you, madam. And your project, the recent one, I did not know, the evaluation of psychiatric disorder in postpartum women, that's an eye-opener. And that every one of us must have come across one or other patient. But then nobody thought about having this, taking it up. And probably in future, she will be taking up antenatal patient having depression. And on top of it, she represents India in international platform. She is ICOG past president, and then a lot of credit and that source. So I welcome you, madam, and then hope you will bless us, and with your blessings, we move forward. Thank you, and over to Dr. Hema, please.
Namaskar. On my own behalf, my heartfelt thank you for all of you for making me a part of this very exciting and insightful brainstorming that this program promises to be. So much that Mandakini has told about me. I have only two things to tell about her. One is that for Mandakini, silence speaks stronger and greater than words. Yes, ladies and gentlemen, she is a woman of few words, but full of action, because just by a chance seating arrangement in one of the conferences, I got to know her more closely, and the kind of volume and the depth of work that she does was really, really mind-boggling. And I was telling her that she must dream big and aim big, and rightly so. I saw on the slide today that she is indeed aiming to start the super specialization in maternal and fetal medicine, the much needed. course for today's generation and the expectations of the patients as well. Well, the second point is Pradhan is her name, like we say Pradhan Mantri. She is a Pradhan of stakeholder of this genetic space, fetal medicine and genetics space, being the chairman of the committee for a prestigious organization, FOGSI, where the leadership always encourages the doers. And she has taken the committee several steps forward in her mission to reach the information to everybody. In this Google era, it's a shame if the patients know more than the consultant. That is what is the common scenario, because it's their problem. They have the time, they have the access to information, and indeed, it is we who have to run and catch up with all that is happening in this space. And I'm very thankful to MedGenome for putting us together for this very crucial discussion. Because when we say, okay, in an event, 20-25 जन तो आ जाएंगे, एक आध कोई आए नहीं तो क्या हुआ? Okay, today is that ekad single gene, which makes a lot of difference. Ek gene bhi nahi raha, though it is going to be, the story of that family, and that person is going to be so different. So the brilliant faculty, which the team, which has been put together for this afternoon's deliberation, with Shagun, Sheetal, and Priya discussing about the single gene disorders, the carrier status, and what we are looking to in the future. If it becomes available to everybody and it is cost effective, yes, the non-invasive tests are going to be the front runners for the frontline healthcare providers. So reaching every girl, every woman is our dream. And the marriage between the medical field and the technology space is really giving us a tremendous potential to do more and more. As you would see from different parts of this great country, we are connected today indeed by the virtual platform, and we have an opportunity to listen to the experts and try and understand better. So may I very fondly wish this program a great success. And Mandakini, I must say, I'm not the guest of honor. I'm honored to be the guest. That is what your and my heart-to-heart connection is. And thank you, Joseph and team, for getting more involved in the knowledge share and dissemination, which is the first big step for people to reflect on how much they know, how much they do not know. The steps further will be to facilitate this knowledge and thinking process into action. And for that, we need a huge framework and a network of small places in the country to the large metros. How do we connect and counsel and take the management in a particular case forward? Well, it's often said that life is full of probabilities, and Corona has taught us that there is always a positive probability. Same is our wish for the probabilities of somebody landing up with a serious issue with the genetic disorders. And I do hope that we understand better and we put this knowledge with our wise action towards the best benefit of everybody concerned. With those words, I would once again very sincerely thank Mandakini Pradhan and the MedGenome team, and wish the brilliant speakers, the faculty for today, who are very accomplished in their own niche, super specialty field, to let the routine OBGYNs, the common man in the battlefield of the genetic disorders, know and learn more. Thank you again. Namaskar.
Thank you, ma'am. Thank you for all those kind words. I can't express. In fact, truly, I can't express in words. And these are people who have encouraged to move ahead in obstetrics and gynecological field. And we do experience these type of problem. Get to see patients. We are lost. And as a FOGSI, the Genetics and Fetal Medicine Committee is committed to it to help our FOGSians. For that, I have sent email for across India, all societies, secretaries and president to be disseminated to all members. If they have any difficulties in solving their cases, they can put into one common email. I've got the whole committee. This email is forwarded to different persons of the committee, and then the response is sent to the concerned OBGYN in 72 hours. That's my promise to all FOGSians. And I must thank Dr. Sagun that she has responded to one of the problem. And I think that more and more such problem are put into this committee, the test of the time, and the committee starts working more. Thankful to Shagun, she promptly within, say, less than 6 hours, she responded, then we have forwarded this, and that is helpful. That is how we can probably reach to the last person. This happens almost every day. I get to see some or other problem and to solve. So I now have a committee. So thank you, ma'am, and thank you all the attendees for this wonderful connectivity. And I hand over to Mr. Joseph to take it forward.
So thank you very much, ma'am, for those words, and thank you, Dr. Hema, for those very kind words that you've given us and that encouragement. So without further delay, now we'll move on to our first speaker for the day. It's Dr. Shagun Agarwal. So she's completed her MBBS from Maulana Azad Medical College, Delhi, and MD in obstetrics and gynecology from University College Medical Science, Delhi, and a DM in medical genetics from SGPGIMS, Lucknow. She is the Associate Professor, Department of Medical Genetics at Nizam Institute of Medical Sciences in Hyderabad, and also the adjunct scientist at Center for DNA Fingerprinting and Diagnostics in Hyderabad. Dr. Shagun is credited with discovery of novel genes causative of human phenotypes HERC1, SERPINA11. She has conceptualized and started MSc Genetic Counseling program at the Host Institute. She is the coordinator for the SIAMG, Society for Indian Academy of Medical Genetics, Clinical Genetics Fellowships, and Genetic Telemeeting. She is the treasurer of the Indian Academy of Medical Genetics and the executive committee member of the Indian Academy of Medical Genetics and Society of Fetal Medicine. She has DHR Short Fellowship recipient in 2018 and holder of 3 research grants. She has several research papers to her credit, both indexed and non-indexed. She has several book chapters also to her credit, and she is also the editor of Genetics Update and Next Generation Clinician. Her research interests are into genetic characterization of fetal malformation syndrome. So without any further delay, I will now hand over to Dr. Shagun and request her to take this forward.
Thank you, Joseph, for that exhaustive introduction. And thank you, Dr. Mandakini, for including me in this program. So today then we are going to talk about kind of much neglected or less famous type of genetic disorders. So these are actually the single gene disorders. So most of all obstetricians are actually quite familiar with the chromosomal abnormalities, Down syndrome, Turner syndrome, and various other abnormalities. But single gene disorders are something which is not that much into the mainstream, and we are not even mostly aware of how to go about suspecting these conditions, how to do testing for that. So I would like to kind of take you through some cases and some basic concepts so that you would have a better idea regarding this group of conditions. So So first, let's just look at some idea about how basically our human genome is organized inside the cells. So all us humans actually have a diploid set of chromosomes, or a double set of our DNA, and this is basically made of 3 billion base pairs. So base pairs are nothing but the AGTC, what you may have heard, adenine, guanine, thymine, uracil. So this is what composes our DNA, and in this long thread of DNA, actually there are certain stretches which code for certain proteins, like which are functional part of the DNA. So these are what we call as genes. And this long thread of DNA actually, to fit into a cell, it has to kind of coil upon itself. So it kind of coils and then supercoils and all, and then it forms these structures which are called as chromosomes. So chromosomes are basically the visible, microscopically visible part of our genome. DNA is something which we cannot really see under the microscope per se. So each of these chromosomes would contain 100 to thousands of genes within themselves, and all of us have 23 pairs of chromosomes in each of our body cells. So with this kind of a basic understanding, now let's try to see what can be the different kind of genetic diseases which can arise. So the group of conditions which most of you would be familiar with are the chromosomal disorders. So these are the conditions which arise because of a change in the number of chromosomes. Some chromosome gets lost or some chromosome gets extra. So the common example being Down syndrome, which is extra copy of 21 chromosome, Turner syndrome, which is one less copy of chromosome X. So here you have something, a kind of a genetic aberration which is microscopically visible. We can see it by doing certain cellular preparations. And so these group of conditions are chromosomal disorders. Then we have a group of conditions we call as copy number defects, where we have a part of the chromosome missing or sometimes extra, but this part is so small that it cannot be seen under the microscope. So this is another category. And then the condition, or basically the group of conditions we'll be focusing on today, are the single gene disorder. So in these conditions, actually the genetic defect can be as small as change in only one base. So out of those 3 billion bases, a single base change could lead to what we call as a single gene disorders. So these disorders, again, the inheritance of these disorders depends on which chromosome that particular gene is located on, and whether mutations of both the copies of the gene are needed to cause the disease, or one copy is enough. So depending on that, we have different types of inheritance pattern of these single gene disorder. But what we need to realize is that actually out of the as of now known 6,000 single gene disorders, if we take them together, their birth incidence is as high as 1%. So this is actually double that of chromosomal disorders. So it's not that these conditions are rare, it's just that they are often missed or we don't suspect them. So that's why we need to learn about this group of conditions also. So you may have seen probably in maybe your genetic colleagues and all draw these diagrams. So these are basically the representation of a family tree, and these are called as pedigrees. So by looking at pedigrees, often we get an idea about what kind of inheritance pattern a particular disease has. So like this kind of diseases where it goes from one generation to next is autosomal dominant disease. These conditions which are found usually in siblings and are more likely to happen in consanguineous couples are autosomal recessive diseases. Conditions which are carried by asymptomatic females in family and affecting mainly males are X-linked recessive disorder. And then we also have some rare conditions which fit into X-linked dominant disorders, and the most severe of them actually present as male lethality in family, and only the surviving female members are there who are affected with this particular group of conditions. So these are kind of the main 4 inheritance patterns of the various single gene disorders. So now let's move on to the clinical aspects. So how would a single gene disorder present to you as a practitioner of obstetrics and gynecology? So these conditions can actually present throughout the reproductive life in the premarital period, preconceptual period, and prenatal period with various different presentations. So in the premarital period, it could be in the form of menstrual problems, genital ambiguity, or sometimes they may come because of consanguinity or family history of genetic disorders. In the preconceptual period, again, you can have these various issues for which the patient may present. And in the prenatal period, which probably you would be more familiar with as being associated with genetic conditions, you could have various scenarios, couple coming because they are consanguineous, or couple with a family history of genetic disorder, recurrent pregnancy losses, previous child with a genetic disorder, or an abnormal ultrasound findings. So these are the various clinical scenarios you would come across. So let's now just go through each of these scenarios and see in what way some of the single gene diseases could present and what we can do to diagnose and manage them. So this is the first case of a 21-year-old unmarried girl, and she is born of a consanguineous parents, and she is basically presented with the clitoromegaly and excessive pubic hair right from birth, with increased virilization and precocious puberty starting at around 10 years of age. No history of any salt losing crisis. Unremarkable clinical examination, normal karyotype, but we can see here the very high 17 OHP levels. So basically her clinical picture is suggestive of a non-salt losing type of classical congenital adrenal hyperplasia. So we know that this particular condition is an autosomal recessive condition, and it is caused by mutations in the CYP21A2 gene. So that's why here we went ahead to do the molecular testing for this lady. So in such a scenario where you have a single clinical diagnosis and that particular condition is caused by mutations in one gene only, so then we can employ something called as a Sanger sequencing. So that is what was done for this patient, and she was found to actually have a homozygous mutation in CYP21A2 gene. So what do we mean by Sanger sequencing now? So Sanger sequencing is basically the gold standard of molecular diagnosis, in fact. So this is a technique whereby you would take a particular gene and you would kind of read the sequence of the gene from starting to end. So you would know exactly in that particular gene what is where is A, G, C, T, in what sequence and order they are coming, and then you can compare it with the reference information what we have, and then you can come to know whether there is any mutation or a change in that particular gene in that patient. So this is the technique which we use in situations where we have a specific clinical diagnosis and we know which gene we want to look for to check for the mutation. So in this scenario, like what this patient had, so when you have genital ambiguity or other DSDs, so we all know, of course, that chromosomal abnormalities are one of the major causes, but when we also need to realize that, especially for a female, there could be many single gene conditions, one of them being CAH, and then there could be certain receptor defects like androgen receptor defects and various other monogenic forms of sex reversal. So all these single gene disorders can also present with the similar kind of a presentation of a DSD, and it is important here to make a genetic diagnosis because it does help in management of the patient. For example, now this girl, once we have made a diagnosis of CH for her, we can start her on steroid therapy, and this would actually help in reducing her hirsutism, and once she gets married, it would also help her in having a spontaneous pregnancy. Otherwise, she would end up into a PCOS and infertility. So making a diagnosis is really important, and even for other conditions, once a diagnosis is made, you can take decisions for further management. Other important aspect here is to realize the recurrence risk. So because this is an autosomal recessive condition, there is a 25% risk of recurrence in the siblings. So in such cases, we could also offer the prenatal testing as required to the at-risk pregnancies in the family. So now let's see a second case scenario. So here we have a 28-year-old lady who's planning to get married, and she's presented with secondary amenorrhea, and basically her hormonal profile shows a hypergonadotrophic hypogonadism. But the interesting point here is that she has two brothers who are both affected with intellectual disability. So these are the photographs of the brother, and you can appreciate in the elder brother a very prominent jaw and prominent looking ears. So actually these two children are having what is known as Fragile X syndrome. So Fragile X syndrome is basically a triplet repeat disorder. So this is also inherited it's a single gene condition, but the molecular mechanism is a triplet repeat expansion here. So here then we went ahead and we did this particular test, which is called as a TP-PCR test for the FMR1 gene, and we confirmed that the brothers indeed had a triplet repeat expansion. So both the brothers were affected with Fragile X syndrome, and what the patient had was actually what we call as a premutation. Like she had an increased number of repeats, but they were not beyond a particular threshold. So this kind of patients are called as premutation carriers. So these premutation carriers are actually at risk of developing premature ovarian failure. And if we look overall in patients with POF who are having a positive family history of POF or intellectual disability, at least 15% of them have this premutation, and even 2 to 5% of sporadic POF patients have a Fragile X premutation. So, but why do we need to find this out? We need to diagnose it because this particular lady, tomorrow if she is able to conceive using her own ovum, either spontaneously or by assisted reproduction, then her male offsprings would be at risk of developing Fragile X syndrome. So we need to offer her pre-implantation or prenatal genetic testing as the situation may be. And other important thing is that even her sister, so she has a younger sister, so the younger sister could also be a Fragile X premutation carrier. So then we could actually do an oocyte retrieval for her before she goes into POF, and this can be help to achieve fertility for her. So again, making a genetic diagnosis here in this patient with POF is important for her management. So now let's move on to a preconceptual or an early prenatal period, where often couples come because they perceive that there could be a risk of a genetic disease in their offspring due to various reasons, most commonly because of a family history. So here we have this 50-degree consanguineous couple, the lady already being 9 weeks, 2 days pregnant, and we can see on her maternal side we have these multiple male members. All of them are having a neuromuscular disorder followed by early death during adolescence. So, and these are all connected through asymptomatic females. So this kind of inheritance pattern is actually quite classical of an X-linked recessive condition. And in such situations, basically all the females in the family are basically carriers. But here we need to now find out whether this lady is also a carrier or not. So how do we go about this? So one more thing we need to realize is that this kind of clinical presentation is actually quite typical of a specific neuromuscular disorder called as Duchenne muscular dystrophy, which shows X-linked recessive inheritance. So here in this pedigree, actually, this lady can already be assumed to be obligate carriers because they are connected through two affected male members. But we cannot assume the carrier status of our patient's mother and the patient. So here what we now do is that actually we're not able to do testing for this lady. So we went ahead and did testing for the patient's mother to see whether she is a carrier or not. So here in case of DMD, the test we use is actually another different kind of molecular test known as MLPA. And this is done because in DMD, the most common mutations are actually deletions or duplications. So here we cannot use Sanger sequencing to make a molecular diagnosis. So in this case, we use this particular technique to test the patient's mother, and she was found to be carrier of deletion of exon 46 to exon 55 of the DMD gene. So the consultant's mother or the patient's mother is now a carrier. So now next step is to check whether the patient is a carrier or not. So fortunately for this patient, she was not a carrier. So because she's not a carrier, now there is almost negligible risk to her pregnancy for her baby to be affected with DMD. So we were able to counsel her and therefore she did not really require prenatal diagnosis. So, so whenever we have a family history of a genetic disorder, then it's very important first thing to make a 3-generation family tree kind of a thing. So what we call as a pedigree. So many times using a pedigree, we are able to get an idea about the pattern of inheritance, and then we are also able to see which are the people in the family who may be carriers, whose children may be at risk. And then using the clinical picture and other testing, often we may be able to also come up at a presumptive clinical diagnosis. But then this we cannot offer prenatal testing on the basis of this information at all. So what is very important to realize is that always a definitive molecular diagnosis has to be made for ideally for that affected person in the family, and then only we can actually identify who's a carrier in the family, who's not a carrier, and then only we can offer a prenatal testing. So without making a diagnosis of an affected individual in the family, offering prenatal diagnosis is not possible. And the choice of the test, of course, depends on what the particular disease is. So in DMD, we use MLPA. In thalassemia, we often use Sanger sequencing. So it depends as per the particular disease and what kind of mutations cause that disease. So now let's move on to another scenario. So this would be a prenatal period where the most common suspicion of a genetic disorder arises when we have an abnormality on ultrasound. So again, let's see a case. This is a consanguineous couple. They already had a preterm neonatal death because of high drops, and now 20 weeks pregnancy. Again, there are findings of high drops. So non-immune high drops we know can be because of various etiologies, and in fact more than 100 single gene disorders can also cause this particular phenotype. So here the baby's karyotype was normal. There was a positive family history, so we are strongly suspecting a single gene disorder. So autopsy here gave us an important clue. So the baby actually showed hepatomegaly, so we suspected a storage disorder, and indeed when we did an enzyme assay, we found that the baby was having deficiency of beta-glucuronidase enzyme, which led us to a diagnosis of mucopolysaccharidosis type 7. And this being an autosomal recessive condition, now prenatal testing can be done by 11 to 12 weeks using CVS. So similar to high drops, we can have various other sonographic findings which indicate an increased risk of genetic disorders. So we have the various soft markers which mostly increase the risk for chromosomal abnormalities. We can have various miscellaneous abnormalities, sometimes because of genetic condition, but the most important group are the structural abnormalities. So whenever you have a structural abnormality in the ultrasound, this could be part of a chromosomal abnormality or could even be part of a single gene disorder. So overall, like the 2 to 3% live borns who have a structural abnormality, this at least 15 to 35% are actually because of a genetic condition, and chromosomal abnormality and single gene disorder contribute equally to this. So it's important not to forget the single gene disorder whenever you see a structural abnormality on ultrasound. So just to take an example of a congenital heart disease. So congenital heart disease could be isolated, but could be part of a chromosomal disorder like Down syndrome, could be part of a microdeletion syndrome like Dijon syndrome, or could even be part of a single gene disorder like Noonan syndrome. So all these three group of genetic disorders actually need to be ruled out before we say that a congenital heart disease is isolated. And it's important to diagnose these genetic conditions because they have other associated problems in the neonatal period and subsequent life. So similar to then cardiac defects, high drops, various other ultrasound abnormality, besides having a significant risk of chromosomal abnormality, can also be associated with copy number defects and can also be associated with various single gene disorders. So it's important that we should not miss any of this. Many of these conditions, that a patient will be having intellectual disability or other problems postnatally. And the other important thing is that unlike chromosomal disorders, for single gene disorder, the recurrence risk is quite high even for subsequent pregnancies. So that is another important reason why we should not miss it. So it's important that you should definitely think beyond doing a karyotype or a microarray whenever you are seeing an abnormality on scan. So let's just see this through some cases again. So we have this particular couple consanguineously married. The lady comes in the 17 weeks of pregnancy and they have a previous child who had died in the infancy FOGSI and had corpus callosum agenesis, developmental delay, seizures, and other problems. And now at 17 weeks itself, we can start seeing early ventriculomegaly features. So at this point, a karyotype was done, it was normal, and an MRI was planned. And that MRI indeed confirmed that this baby again had a corpus callosum agenesis. Now, because of the postnatal phenotype of the previous child, we were able to counsel them that this is likely to be poor prognosis. So they terminated the pregnancy. Autopsy did not show any other finding except for a corpus callosum agenesis and some abnormal cortical layering on histopathology of the brain. So with this kind of a scenario now where we have two babies having the same kind of finding and a couple being consanguineous, so we are strongly suspecting this is a single gene disorder, but we are not really having any clue as to what it could be. So now how do we find out what is the single gene disorder this particular family is affected with? So there are overall more than 6,000 genetic single gene disorders, as I told earlier, and conditions which present with corpus callosum agenesis are around, on an average, around 100 different single gene disorders. So now what do we do? How do we find what is the condition in this family? So in such a scenario, we have basically technology which comes to our rescue. So as we saw earlier, if we want to do, if we want to use Sanger sequencing, we actually need to narrow down to a single diagnosis, a single gene. So that is not possible now in this particular case. So what we do here is what is called as an exome sequencing. So just to give you an kind of an understanding of what do we do here or how does it differ. So when we do a karyotype, so it's like, you know, looking at Hyderabad where I stay. So it's kind of looking at Hyderabad from a zoomed out view. So this is a kind of a zoomed out map of Hyderabad. We can see some main features, the main famous areas of Hyderabad, but nothing beyond that. So a karyotype is limited in its resolution. It will see only some major things like a missing chromosome or an extra chromosome. When we do a microarray, we are able to zoom in a bit and we are able to see certain more important features within the map here. So some other important regions are coming up now. So a microarray is basically going to show us some other kind of genetic defects, what we saw earlier, the copy number defects. So this is what comes up in a microarray. But when we have a single gene disorder, then what to do? So when we are looking, we want to look at one gene, so we do Sanger sequencing. So this is like looking at the Charminar, which is one of the iconic monuments of our city. So it's like looking at Charminar in great detail. But what if I want to look at all the genes or at least 6,000 genes which cause disease? So then is what comes up, the test which comes up is next generation sequencing. So next generation sequencing is something whereby actually we are able to sequence all the genes which are there in the DNA in one single experiment. And for this, the clinically relevant technology is what is called as an exome sequencing, or more commonly the clinical exome, where we sequence all the known disease causing genes. So when we do an NGS, or when we are doing basically a clinical exome sequencing, it's like akin to kind of seeing a Google Earth picture now of a little zoomed in map. So we are actually able to see everything, the whole of Hyderabad, and we are able to see details of small locations also. So here we are now going, the resolution is going for a single base level, but throughout the genome. So that is kind of the marvel of the technology, we can say. So here in this patient, then what it showed us that the baby actually had a homozygous strain shift deletion in this EPG5 gene, and this led us to a diagnosis of a relatively rare condition called as Vicky syndrome. And then when we reviewed the records of the previous child, so that child's clinical phenotype also was consistent with this diagnosis. So this being an autosomal recessive condition, the recurrence risk was 25%, and we were able to provide them prenatal, and they had a healthy baby subsequently. So the point here to remember is that on scan, the only finding here was isolated corpus callosum agenesis, and that is one of the common things which you come across when we during ultrasound. And this usually has a normal postnatal outcome in more than 70% babies. But we need to remember it may be part of a genetic syndrome, chromosomal condition, copy number defect, or even a single gene defect. And there, those children can have an intellectual handicap. Thus, we need to rule out all these conditions during the prenatal period. even something like a liker abnormality, which are usually because of acquired factors. So here this family, second degree consanguineously married, had 2 babies with severe anhydramnios with no obvious cause of the anhydramnios, no maternal condition. Again, the autopsy, histopathology, everything was non-conclusive. Karyotypes were normal. So suspecting an autosomal recessive condition, we did an exome sequencing, and this particular baby was actually having a homozygous splice site mutation in the ACE gene, and this causes autosomal recessive renal tubular dysgenesis. So we were able to give them prenatal in next pregnancy. The baby was luckily heterozygous. So heterozygous means that the baby was a carrier and not affected. So subsequently, this is the baby which was born. So the patient lived in a far off village. So he sent the photograph of the baby after birth. So again here, like as I said earlier, so liker abnormalities, we don't really think of genetic condition, but here because of a positive family history and no obvious cause for the anhydramnios, we had a very high index of suspicion, and this was confirmed. So if we see the literature now, exome sequencing has actually been used for ultrasound abnormalities and also autopsy abnormalities over the last 4 or 5 years, and we have now multiple papers and even now recommendations coming from ISPD, SFM as well as ACMG, and all this literature basically shows that at least 20 to 30% of cases where we have a structural abnormality in a fetus, this could be because of a single gene disorders, and these really need to be diagnosed, and they also have a very high recurrence risk in subsequent pregnancies, in contrast to the chromosomal disorder where the recurrence risk is usually low unless one of the parent is a balanced rearrangement carrier. So doing an NGS in the scenario of abnormal ultrasound finding really helps us in making a diagnosis of a single gene condition, and if they are a poor outcome condition, then the patient can take a decision for termination. In conditions where there could be a treatment available, that can be planned beforehand, and of course it's important for recurrence risk counseling and prenatal testing in subsequent pregnancies. But it's important to recognize that this testing at present is challenging. It has causes a lot of problems in the data interpretation, so it should be done in consultation with a genetics professional, and a pre-post test counseling and informed consent is important. So just to move on to the last scenario of recurrent pregnancy loss or a bad obstetric history, again a consanguineous couple, multiple first trimester abortions, but interestingly 2 second trimesters IUDs also, and this baby had some hydrops also. So this is the autopsy findings of the baby, some subtle dysmorphism, but what is interesting is a very unique kind of cardiac defect. So this is actually a rudimentary right ventricle, what we can see here for this baby, and this is a kind of a nubbin on the surface of that, and along with that there was a pulmonary artery stenosis. So basically it's kind of a hypoplastic or atretic right heart for this baby. Microarray was normal. So again it was exome sequencing which led us to the diagnosis. So the baby actually had a homozygous mutation in the PLD1 gene, and this is known to cause a right-sided cardiac valvular developmental defect. So this was quite consistent with our findings, and then we did a segregation in the family. So again, this being an autosomal recessive condition, so we can counsel them for recurrence risk and prenatal testing. So again, recurrent pregnancy losses or bad obstetric history, we are familiar with chromosomal abnormalities, and most of you, I hope, are ordering karyotype of the couple, but we also need to remember that single gene disorders can also present like that, and to recognize this, it is important to do an autopsy evaluation, and all pregnancy losses beyond 12 weeks of gestation, even if it's a single loss, should undergo an autopsy, and if you're not able to do that, at least take photographs, radiographs, preserve the records, and most importantly preserve the DNA of the baby, which can be done through all these various samples, and then subsequently you can consult a geneticist and they can advise you for the further genetic evaluation. So in this family, in fact, we had another common scenario which was of a recurrent neonatal death. So 2 previous neonatal deaths, and now again this baby was admitted in the pediatric hospital with encephalopathy, and the father had come to us with the samples of the baby, and what we found was basically clear very high glycine levels in the plasma of this baby. So this was consistent with a particular genetic condition called as non-ketotic hyperglycemia, which is again an autosomal recessive condition. But unfortunately, before we could get the DNA of the baby had expired. So here we did the sequencing of the parents, because they would be carriers of this autosomal recessive condition, and we were able to find the mutation in this family, and we were also able to offer them prenatal testing, and they had a healthy baby subsequently. So again, for neonatal deaths, we need to remember this could also be because of single gene conditions. Many times it's an inborn error of metabolism. Sometimes it could be a lethal skeletal dysplasia or other syndromes. So it's very important to suspect this, to preserve photos, X-ray, clinical findings, ideally do an autopsy, and also to preserve the sample of the baby, because even though many times a couple are not ready for a genetic testing at that point, they may be ready subsequently when they want to plan another pregnancy. So it's very important as doctors to tell and counsel these families to save their sample, so that subsequently a genetic evaluation can be done. And finally, the last thing, so whatever we have seen till now is a diagnosis of a single gene disorder and a kind of secondary prevention. But it's also important to remember that we can provide primary prevention of many single gene disorders. So Dr. Sheetal would be subsequently talking in detail about it. So just to mention the most common genetic disorder for which carrier testing is easily available in our country, this is thalassemia. So 10% of Indians on an average are carriers of thalassemia, and these carriers will not have any symptoms, and any two carriers can get married to each other. And once these people get married, then there is a 25% chance in their every pregnancy of the baby having thalassemia major. And this can easily be prevented by doing a carrier testing for every lady who is planning a pregnancy. This can be done by a simple cheap biochemical test called as HPLC. And when both partners are carriers, then we need to do a genetic testing to find the mutations, because then only we can offer them prenatal testing. So to conclude, then we need to realize that single gene disorders can present in various ways at different periods in the reproductive life to an obstetrician. And as an obstetrician, in fact, you are very uniquely poised, because you are the one who can prevent the birth of a child with a genetic disorder. None of the other physicians can do it. You're the one who can actually act and prevent these conditions from coming. So it's important to realize that you should be able to identify these individuals and family who are at risk for this condition, do an appropriate test which should not be limited to a karyotype, consult a geneticist if you are not sure of your diagnosis and you need to understand what further testing is required. So workup for single gene disorders is important, of course, as we have seen, and the choice of the test often depends on the disease. And making the diagnosis, besides helping the patient in management, helps in finding the recurrence risk for the family, helps in providing prenatal testing. And whenever we have a positive family history, it's very important that it is the workup of the affected person which needs to be done. We cannot do any testing on the couple who has come to us. It's the affected person who needs to be tested, and the best time to doing this workup is the pre-conception period. And now we of course have various novel techniques like the next generation sequencing, which can provide us answers in cases where previously we were not really able to come to any conclusion. So I think with this I will stop. Thank you.
Thank you very much, Shagun. I think we have just one question.
Whether trisomy 21 can be a cause of DSD?
Yeah.
No, if the question is regarding a newborn probably. So in such a situation, trisomy 21 per se doesn't cause a DSD actually. So, but it could always be a coexisting diagnosis for that child. But DSD is not a presentation of trisomy 21.
Okay.
There was one more question actually, and by mistake I only deleted, answered it, answer live. I just pressed that button. The question was, if we universally ask for screening of women for single gene disorder, or it is case to case basis. So Dr. Sagun, you can answer.
Yes, so I think Dr. Sheetal would be probably discussing about that expanded carrier screening. So for carrier screening, as of now, the recommendation per se is to screen for the condition, one which is common in the population. So for us Indians, it is basically beta thalassemia. So 10% of Indians are carriers on an average for beta thalassemia. So we need to do carrier testing for thalassemia for every couple. So that is one on basis of population prevalence. And the other thing is it needs to be individualized depending on the family history. So it's always important to take the family history, and then if there is suspicion of a genetic disorder in that particular family, then we, as I said earlier also, then we first need to make a diagnosis of that affected person, and based on the inheritance pattern and all, then we come to know whether the lady needs a carrier testing for that condition. So that would be the individualized carrier screening depending on a family history. So, but as far as expanded carrier screening for all genetic conditions is concerned, as of now, it is not really recommended, but it can be done. So I think Dr. Sheetal would be discussing the pros and cons of that in her talk.
Dr. Shagun, there is somebody is asking how to store blood or skin tissue of an affected child who has expired or it is IUD.
Okay, so like if we are looking at DNA banking, so we can store the skin, we can do it from either the blood sample, or this blood sample like can be taken as a cord blood during the delivery process, or it could be taken, an intracardiac puncture can be done immediately after birth and the blood can be taken. So this blood needs to be stored in an EDTA vial, and this can be stored in the refrigerator for a short time, maybe like a few up to a week at 2 to 8 degrees. But beyond that, if you need to store it, then you need to actually extract DNA and store it at -20. That is the ideal thing. So for that, you can take help of any lab who is able to extract DNA and keep the sample for you. And besides blood, the other samples which can be stored are skin. So you can take a skin biopsy, or if that is also not possible, you can even store the umbilical cord. So skin and umbilical cord can, again, they can be put in normal saline and stored in the refrigerator for a short while, but preferably then DNA should be extracted, and then once you extract DNA, it remains stable and you can store it long term at -20 degrees. And yeah, so and one more thing, like which is not that prevalent in north but actually quite prevalent in South India, which helps us many times, is that many patients actually they store a fragment of their cord at home, like they just dry it in the sun and keep it with them. So that actually helps many times. So in fact, here many times when patient families come and they have a previously deceased child, we always ask them whether they have stored a cord segment. So that also can come, we can actually extract DNA, but that's not preferred. So if you have an opportunity as a doctor to store the sample, so whatever I mentioned earlier, those are the ways in which you can store the sample of the baby for further evaluation.
Okay, I think we'll move on to the next speaker in the interest of time and come back and address all the questions. So our next speaker is our own Dr. Sheetal Sharda. So Dr. Sheetal is a pediatrician and has done a DM in medical genetics from SGPGI Lucknow. She has worked as an associate professor in genetics at PGIMR Chandigarh for 8 years. She has over 14 years of experience in clinical genetics, and she has over 30 publications in national and international journals to her credit. Her areas of interest are in dysmorphology, prenatal diagnosis, and molecular diagnosis. I hand over to Dr. Sheetal and request her to take it.
Hello, good afternoon, everyone. At the outset, I would like to thank Dr. Mandakini ma'am for getting us all together. And I'm not sure whether she remembers, but the first time I took a lecture on carrier screening was in one of the events she had organized in Lucknow. And we have come a long way since then, from 2017 to 2020. And I am seeing the changes which are occurring due to the increasing knowledge, awareness among the clinicians, as well as the general population, as they know what's going on and they are demanding that. So I thank ma'am again, and I also thank MedGenome, Dr. Priya and Joseph for always making efforts to bring us all on a same platform, since we are all working in reproductive genetics. So a combined effort is always recognized because they all are interlinked, right? So one is not effective independently. And I would also like to thank Dr. Shagun for making my work so much simpler, because once she has explained what single gene disorders are and what tests are available for doing the test, it becomes easier to explain to them, to everybody, that how we can do carrier screening for single gene disorders. It is an upcoming topics. Many things are not clear. There are many gray zones. But as other things evolve, probably carrier screening is also a thing which is evolving, and soon it will be recognized as one of the important steps in genetic counseling during reproductive decision making. So taking that, I'll start my talk. So first of all, I would just talk a little about what's the background of carrier screening and how it is relevant for Indian perspective, because whatever literature we search, it's always what has happened in somewhere outside of India, and sometimes we are not able to reciprocate the same in our Indian scenario. Secondly, I would talk about expanded genetic carrier screening. Targeted screening, already Dr. Shagun has showed you a few cases, example in thalassemia. And more importantly, since we don't have any current recommendations or guidelines in India, we as people who work for evidence-based medicine should have a rational approach, and can we decide on our own basis that what would be the best step forward. And obviously, we need to have certain ethical considerations when we are offering carrier screening as an option to couples. Okay. So we all know that genetic disorders are diseases with a burden, and they are obviously familial conditions, and most of them would not be treatable. So a couple would always come to you in scenarios where they have one child affected with a genetic disorder. They have lost previous children because of genetic disorder, or the person himself is probably having a genetic disorder and they want to know the risk in their offspring. Or like Dr. Shagun explained the example of DMD, they need to know what is the risk in their offspring. And sometimes it may be just a couple who is just planning pregnancy or recently got pregnant, but they know that because of consanguinity, they are at a higher risk compared to the general population for a child having a genetic disorder. So these would be various case scenarios in which they would try to understand what is their risk. So prevention of genetic disorder is the only way how we can decrease the burden, because as we all know, majority of them do not have a curable or a treatable option for them. So current status is either the child is already born and we are able to take care of the complications, right? So that is what is known as tertiary prevention. So we are just managing the complications. There could be secondary prevention in which we know that the previous child had something which can be detected very early, and before the symptoms appear, if we diagnose, then we might have an option of giving some form of management or to decrease the complications which can occur. Or now we have the options of primary prevention, and this would be possible only when we have already one child affected and we want to prevent it in the next child. So here we can either use prenatal diagnosis, or we also now have the option of a pre-implantation genetic diagnosis, and possibly in near future for single gene disorders, NIPT may also be an option. Dr. Priya can talk about it more, but probably we'll have to wait for it a little longer. So in all these situations, where does carrier screening come? Can it be used for primary prevention before the birth of even the first child? So this is how carrier screening can be implied for single gene disorders. So carrier screening is not a new thing. It has been happening all over the world. And there are certain committees which have their definitions or opinions. And basically it is that a test which is performed on an individual who does not have any overt phenotype for a genetic disorder, but may have one variant allele which can be associated with a genetic diagnosis. So in simple terms, it is a screening test. It is not a diagnostic test. It is a screening test done to know whether the unaffected or the asymptomatic parents have any risk of having a child with an inherited recessive disorder. So why I am focusing on recessive is that if we are testing a couple or partners, they both have to be having this variance in the same gene or alleles to have a autosomal recessive disorder, or the mother is a carrier of an X-linked recessive disorder, though we can surely do it for autosomal dominant disorders if one of the spouse is having a disorder, right? So carrier screening is basically done in asymptomatic population. So what are the recommendations? I think this was the question which was asked by Dr. Ashish Faujdar. So they are recommendations, but again, it is American College. In India, there are no recommendations. It is said that the carrier screening information should be provided to every pregnant woman, ideally before pregnancy. But as we all know, we are often recruited only after the pregnancy is there. So if one of the partner is found to be a carrier for a specific genetic condition, then the spouse has to be offered the genetic testing. And if both the partners are found to be carriers, then we have to offer them counseling so that they can take an informed decision. And in all these recommendations, they emphasize that they have to have a family history, and the patient should also be encouraged that they may be carriers, but they have to also inform their other relatives who would be at risk, and they also have to be offered a carrier screening. So these are the recommendations. So the purpose of genetic carrier testing is preventing the birth of a child with a genetic disorder which is lethal, untreatable, with high morbidity and mortality. So these are the disorders which currently we should be focusing upon. This again I'm telling because nowadays they are also coming forward to do carrier screening for complex disorders or polygenic disorders like diabetes. So currently there are no recommendations, and we all know that these are just susceptibility genes or they are just predictive genes, and having them doesn't mean that there is 100% chance that the person will have the disorder. So the penetrance is not complete, or there are multiple genes which will be involved. And for those disorders, ideally, currently, we should not be offering a carrier screening. So when we see the background that why we are offering carrier screening is that every individual, each of us harbor an average of around 3 severe recessive mutations. Severe in the meaning that they could be nonsense termination variants. So each of us, even if we have no family history and we are just doing a general screening, we might be carrying 2 or 3 recessive genes which are pathogenic or likely pathogenic. And these couples, unfortunately, they may not be even aware that they are having this genetic variant, and they might have a child, and only after such an affected child has been born that they are known to be carriers, just like in thalassemia major. So carriers are usually asymptomatic and they have no family history. And right from 1984 to a recent study in 2017, the percentage is almost remaining same. That almost 80 to 90% of the carriers had no family history of cystic fibrosis, and these children were born with autosomal recessive condition, which is cystic fibrosis. So asymptomatic, no family history, so we don't have any high risk group whom we can focus that, okay, you have to be screened for certain disorders. Secondly, when we look at the Indian perspective, we all are aware of the high population, high birth rate, as well as a high consanguinity. So almost in certain population, the consanguinity can go as high as 40%. Some communities like uncle-niece marriage is a norm. So in such situation, we can understand that the impact of the genetic disorder burden is very, very high. So keeping this in mind, we know that there have not been many studies for genetic disorders in India. And whatever studies we have done are based only on certain small studies, newborn screening, which shows that we have a very high carrier frequency of common genetic disorders like galactosemia, CAH, thalassemia, and congenital hypothyroidism. So based on this, we have to ask the question, Is there any need for genetic carrier screening? And I think you all would agree that there is definitely a need for doing a genetic carrier screening. So now coming to the rational approach, since we don't have recommendations, whom would you approach for doing a carrier screening? So whether you would only focus on high risk or ethnicity based population, or you want to go forward for an expanded universal screening. And how would you do the testing? Do you want to do targeted screening, or do you want to do expanded screening by next generation sequencing? And when will be the ideal time? So whether periconceptional is good, preconceptional is good, or premarital is good. So when we compare traditional and expanded, traditional is always based on ethnicity. But we understand that in India, it is more of a multicultural population, and so ethnic distinctions have become less. Worldwide also, first, which were ancestry-based testing, now they have evolved and they are going to pan-ethnic testing. So now there are very limited places where we are doing ethnicity-based testing. So this was a very good study. It is a hypothetical study, yet they have compared that if you do traditional screening is you take a cohort of people based on ethnicity and you do the common mutations in them, versus expanded carrier screening, in which you ignore the ethnicity, you give a generalized carrier screening. And they found that even hypothetically, if they are doing the traditional screening, they can identify around 55 affected fetuses. Compared to the expanded carrier, which can detect almost 3 times more affected fetuses. So we can understand that the traditional screening is now getting obsolete when the next generation sequencing has come up to help us in doing the expanded screening. So in India, what is the status in India is that we are presently limited to common genetic disorders. So we do have many genetic centers. We have awareness for common diseases like cystic fibrosis or hemoglobinopathies or hemophilia, DMD. So especially means that only in couples with a family or a sibling history. So this is a limited carrier screening. But India is a heterogeneous mix, right? So we know that there are pockets of heterogeneous, though it is a heterogeneous population, there are certain areas which are homogeneous, like the tribal population, which will have more common in sickle cell. We often ask history whether you are coming from Sindh or Punjab, and we know that there are certain common thalassemia mutations which are more common in those communities, and E beta is more common in the Northeast population. So based on community, if we say, then there are homogeneous population. So they could be dependent on the inbreeding, the consanguinity, or caste-based testing. But unfortunately, we don't have population-based data. So we cannot focus based on ethnicity. So even in India, if we focus on ancestry-based, then it will lead to an unbalanced distribution of the genetic testing. So now CST has, or carrier screening, has moved from targeted mutation to next generation sequencing, in which we can test for multiple disorders together, just like Dr. Shagun had mentioned, that either you do by Sanger sequencing, but that would be focused. So we have to know what disorder we are checking, and in that we will do the carrier screening. Compared to disorders in which there are multiple genes or multiple phenotypes associated, or a varied type of genes and phenotypes are evolving, then Sanger sequencing becomes a little less informative. So we will have to go for NGS. So even in the West now, they were using NGS for targeted genotyping because they have very well characterized population-based studies, and they know which are recurrent mutations or which are founder mutations. But again and again, we say the same thing, that we have not done a population-based study for our country, so we cannot focus on targeted genotyping even by NGS, let alone by Sanger. So NGS is definitely a better method now to check for common mutations, rare mutations, as well as novel mutations. And so we know that NGS can be much more helpful in giving you a result compared to a targeted testing. And this is all because of the advances in molecular testing. You have heard, Dr. Shagun, that right from single gene to triple repeat disorders to point mutation to copy number variations to karyotype, many of these technologies are evolving on a better scale, and diagnosis is becoming easier and faster and also cost effective. The tests which were very costly initially, like 50,000, 40,000, now you can do it even at 15, 20,000. So since now we don't have that reason that the tests are, we are not having tests or the tests are very costly and so we are not doing it. It is just that for what disorder you have to choose the right test and it becomes cost effective. So the next question comes is, which is the ideal period when to test? So better timeline obviously would be preconceptional, that we have enough time for a detailed workup of the family. Either the siblings were affected or previous offspring was affected, they are not with us. So we can have enough time for a detailed workup, and if something positive or negative is there, they can take an informed decision. But probably most of the couples come periconceptionally. And here it is easier to offer them the carrier screening because it is a vulnerable period and they're very receptive to the test. Okay, they have a strong family history, so they will be ready to accept that test and do it. Certain other options are premarital carrier screening. This seems a very off the record thing, but I'm seeing the difference that when we started in 2017, till now, the number of cases which we are getting for premarital carrier screening based on strong family history or consanguinity is increasing. And the other option could be pre-implantation genetic screening of a donor. So these are also discussable options. There are no recommendations or guidelines. But if a couple is already going for an IVF for some disorder, for infertility or something, and we use a donor, we might have a chance of bringing in a disorder which is not there in the family, just because the carrier, the one of the spouse is a carrier and the donor by chance is also a carrier of the same disorder. So this can also be avoided if we do a pre-implantation genetic screening. But for premarital, we all are aware of the social issues in India. So the next question would be, should I marry him or her because she or he is a carrier? So this is a very difficult question to answer because there are two things. In genetic counseling, it is always non-directiveness. We cannot say, yes, you marry or don't marry. We can just give the facts and figures to them. But you have a responsibility as a consultant also, because the consultant is dependent on you for an answer. But as a doctor also, you know that carrier means asymptomatic and they are at no risk themselves. It is just that to the offspring, there is a risk. And if it is a disorder which can be tested by prenatal or by PGD, then it is not wrong to marry, or, you can always have a discussion on that, but it will create a stigma. And we all are aware that in India, still today, the female is always made to feel guilty, even if she is just a carrier. So these social issues and stigma towards a carrier have to be taken in mind. And this all should be considered as pre-test counseling. So you have to counsel the options. We do get counseling for this, but they also want that the party should not know that we are trying to extract information from them, how to get sample from that affected family's person who is in question. So these all limitations are definitely there. So I think this may be again a area where we can have a debate or recommendations later on. So we know that there are many challenges when we are counseling a carrier. So there may be strong religious beliefs, some religions that they will not go for an abortion, or they may not terminate a baby. In some, they don't even want to do a PGD. Some will say, oh, we have a timeline of 120 days. There are so many strong religious beliefs also which comes into pre-test, post-test counseling. So And also the options after doing a carrier screening, what options can be given, even in that they have to have a viewpoint. So all these limitations in carrier testing also should be discussed, and we have to be aware. So I'm just quickly going through few cases where expanded carrier screening, expanded means doing a clinical exome or a whole exome of the couple, has helped in individual cases. So this was a very recent article from Sir Gangaram Hospital, Dr. I.C. Verma and group, and they have studied a group of carrier screening couples and individuals, 200 of them, and this has definitely brought out unexpected results. So there are certain things which we felt were not very common in India, and they were more common in Caucasians, but we have found otherwise. So if you see the reference, it is a really good article to go through, and it will probably be a pilot study to do further such testing in a larger population to understand that are we really different from Caucasians, or do we have the same pattern as far as common genetic disorders are concerned. So here this was a family who had no family history of a genetic disorder. So they had come preconceptionally just because of consanguinity. So this couple, they had a family of complex disorders, right? So heart attack, diabetes, stroke, and it was very clearly pre-test counseling was done that these are complex disorders and we will not be informing them, or this was not a test which can be tested by this NGS screening which we are doing, because we have a limited number of autosomal recessive disorders and X-linked disorders which we do. But this couple was found to be carrier of a termination or a pathogenic variant and a likely pathogenic variant in a gene MUT which causes methylmalonic aciduria. As you can see in paternal grandmother, so we had 3-generation pedigree with no family history suggestive of an inborn error of metabolism, but the couple was found to be carrier of pathogenic and likely pathogenic variant. Besides this, the couple was also carrier of a CFTR variant, which is common in both. Now, in case of consanguinity, we assume that both of them would be carrying the same mutation in the gene. But here you can see that, yes, they do have the same variant in the CFTR gene, but different variants in the MUT gene. So presence of consanguinity doesn't mean that they will have common variants. So that's why instead of doing a single spouse first and doing the same variant in the other partner, sometimes might miss this compound heterozygosity possibility in the offspring, and you might get a baby with the disorder in spite of doing a screening. So this is another couple which had come periconceptional, 9 weeks, previous multiple children affected. So this is a very dreadful history which we take. As you can see, 8 children, out of 8 children, there were 5 children who were affected, and only in one they were able to do a test by TMS and GCMS, which is tested for inborn error of metabolism, and they found that the child had methylmalonic aciduria. Only based on this history and consanguinity, the couple was screened and they were found to be carriers of pathogenic variant. Here there is consanguinity, both the couple have the same variants, and it confirms the TMS finding in one of the sibling whose test was done. So this is, I won't say an ideal because we have lost so many kids already, but this is a situation in which getting such result is really, really helpful for the family for doing a prenatal and then continuing the pregnancy or terminating if the fetus is found to be affected. So again, here there is a non-consanguineous marriage, and the cause was not known. So multiple children affected, and they had lost not 1 or 2, but 5 children. And of them, 4 were in the neonatal period, and 1 child had presented with global developmental delay. So now we can see that 4 children were similarly affected, but 1 child crossed the neonatal period. So the possibility of 2 genetic disorder in the same family is not low. Here, the couple was found to be carrier of pathogenic variants in both the disorders, and they could be explained. So here, in spite of being a non-consanguineous marriage, they had both the variants in the gene which were same. So it doesn't always mean that non-consanguineous cannot have the same variant. These are so rare disorders, yet there could be a possibility of ethnicity based or endogamy. So again, another family with two children affected with different condition, two children with thalassemia, again a very common disorder, and two children were suspected to be having meconium ileus at birth and failure to thrive. So they were suspected for cystic fibrosis, and the common mutation in CFTR was negative. So now out of like 4 children, 2 are having CFTR or a cystic fibrosis suspicion, and 2 children are having thalassemia. It is in such cases, it's really important to go ahead for a combined genetic screening, and the parents were found to be carriers of both cystic fibrosis as well as beta thalassemia. Now, if we do a beta thalassemia screen and then just screen the next fetus for beta thalassemia, and the third fetus comes out to be positive for CFTR, so this is how we might have a family who are having more than one type of condition being missed. And besides that, they were also having a termination variant in another gene for which there was no history, but they both had the same variant. So now in the third pregnancy or the next pregnancy which they plan, along with thalassemia and cystic fibrosis, they will be offered to do testing even for MYH3 gene, so that the risk of 25% for this disorder is also prevented. So this is again similarly 2 different conditions in the same couple, and both were carrying likely pathogenic variants which the first child had, or likely pathogenic variant which the second child was suspected of having. Now these are disorders which clinically also we don't keep as a differential diagnosis. So the first child was suspected to be affected with West syndrome, and the second was suspected to be osteopetrosis, and the genotype which we got was totally different from what a differential diagnosis was. So I'm just showing you all the positive cases to emphasize that in such cases where there is a strong family history or a bad obstetric history, how carrier screening together will give the diagnosis. Sometimes just to save money, or when there is enough time, a single spouse sample is sent that, okay, if something comes, we will do it in the couple, because sometimes there may be a none report and we might not get anything because the phenotype is not very clear. Now here this child had died, 2 children had died for due to neonatal respiratory distress in the first 24 hours or 48 hours. So they were suspected with surfactant deficiency, but the wife was found to be a carrier of myopathy genes, not 1 but 2. Now these are uncertain significance because firstly we do not have the phenotype of both the kids who had died, and secondly because there are 2 variants and we really don't know of these 2 which would have been causative of this. So in this case we then suggested let us to the husband, and unfortunately the husband did not have any of the variants of NEB or col6A1. So this, now if on basis of this, if we do a prenatal that okay, this is a consanguineously married couple, so if the wife is having a single head, the probably husband also must be having the same, and let us do the fetus directly, then you may have chance that fetus will also come as heterozygous. But the husband did not have these variants. Then what to do? So here a combined analysis was done, and now we found common variants in another gene which can explain early death, right? So if you have time, then you can go stepwise. But if you don't have time and the couple has come directly during pregnancy, then it is ideal that you do the combined screening. It will save time and you will get more information. So this was a small internal review of a cohort of around 1,000 people, means 510 couples. And the main reason for referring for carrier screening as expected was the previous child has been affected. So almost 75% of the indications for doing carrier screening was previous child being affected. But there were other indications also, like a bad obstetric history or infertility, or just for a premarital counseling, though numbers were very less, just 9 or 10 couples. But as you can see in infertility, unfortunately, we didn't get many single gene variants which can be explained. So infertility would go lower on the indication for doing a carrier screening. And we can see that VUS, you all have started getting reports of NGS, and this is a very gray zone where we get variants of uncertain significance. Now, if we see that based on a good clinical history, when you have a previous child and you have a clear-cut diagnosis, like the case in which one of the child by TMS was found to have MMA, the chances of getting a pathogenic or likely pathogenic variant is quite high, versus a scenario in which the previous child is affected, but we don't know the diagnosis. So here in such cases, variant of uncertain significance will be quite high. And also when there is a negative family history, even in that, the chances of getting a pathogenic, likely pathogenic variant is 25%. So the distribution of variants, whether it would be pathogenic, likely pathogenic, non-report, or variant of uncertain significance, would definitely depend on the indications for which the carrier screening has been done. So we can conclude from these case studies that obviously carrier screening is very beneficial for those couples in which they have a previous child who is affected by a known clinical, known genetic disorder, and we have a good clinical correlation. So the classification of the variants becomes quite easier for the laboratory. Or a bad obstetric history like IUD or recurrent hydrops fetalis or raised nuchal fold thickness, recurrent cardiac disorders. In such cases where we have some finding based on antenatal ultrasound or autopsy, those couples can also be benefited. Or there are conditions in which the person himself is suffering, like for example, deafness or retinitis pigmentosa, then the partner can be screened. So now the question arises, okay, we have done a carrier testing. How is it going to help them? So this is important for planning the family. So the couple may say, okay, we don't want to take any decision. We are aware and we just want to be prepared. So they will be mentally aware that they might be at risk. They may not take any difficult reproductive decision like termination. Some may opt for newborn screening if it is an IEM and a treatable condition with early testing. Sometimes they may opt, most of the times, I would say not sometimes, the most common reproductive decision is a prenatal diagnosis and selective termination. And now the options of doing a pre-implantation genetic testing are also quite open. The most difficult obviously would be a premarital testing. So this is something which is a gray zone again. And as you can see, if I've just taken 25 cases in which carrier screening was done and we have found a negative report, but the most difficult cases would be in which we had found uncertain significant variants, and unfortunately the fetus also has the same variants, then what to decide. So it becomes very difficult. For example, this case has always remained as an example for us that we had found something, but it was uncertain because we had reported it because we didn't know what the previous two neonatal deaths were happened. We had no details of them. So we had reported them as uncertain significance, and it was clearly told that no reproductive decision has to be taken, and they continued the pregnancy. The variant was quite good, 0 minor allele frequency, damaging by in silico prediction, but yet the fetus, in spite of being homozygous, it was continued. And by 28, 29 weeks, recurrent, the mother used to keep calling the genetic counselor that I'm feeling the same feeling which I had when the previous two fetuses were there. And I think even this is affected. And then unfortunately, she shared one report which was not shared with us previously. And profile had been done, metabolite profile, in which it was towards the diagnosis of carnitine deficiency. And it was really, really sad for us that if this report had been shared earlier, probably the classification of the variant would have been different, and we would have taken a, and this was previous to LSCS, and this also was an LSCS affected child. This is one of the cases in which we always have a red flag that what to do. So the ethical principles, being obstetricians and fetal medicine experts, we know the ethical principles as far as the PCP and the TMTP guidelines are there. and we have to restrict such screening tests to lethal or severely debilitating disease and not complex disorders. And VUS is definitely a big uncertainty. So before taking the step of doing a CVS or an amniotic fluid, we have to go back to the lab and see whether there is a change in the classification. Parental studies needs to be done, other affected siblings need to be done, and we have to try to change or try to reclassify that variant of uncertain significance based on as many clues as we can, especially if they are going to take a reproductive decision, because it is a very tricky situation that we don't report a VUS and the affected child is born, or we report the VUS and the unaffected fetus is terminated. So this is a high risk zone, and we have to be able to address this case to case. So to summarize, it is just that we will start providing expanded carrier screening, but we have to understand that whatever test we select has limitations. It is not a guarantee that you are going to get a totally normal child after you go for whole genome sequencing or whole exome sequencing. And if it comes negative, it doesn't mean that you're not carriers of a genetic disorder. It is just that this field is also evolving, and there are a lot of things which we still don't understand, and there could be different etiologies for genetic disorders, and we have to have a follow-up plan. It's not that you do carrier screening, but then who will do the genetic counseling, who will do the prenatal diagnostic testing, what are the options you are going to provide? And we should not have unrealistic expectations. So pre-test counseling, very important for any genetic test. And obviously the treatment is available for few genetic conditions, and sometimes but overall, by doing carrier screening or prenatal screening or newborn screening, the therapeutic options which are there are not going to improve. It is just that it can be taken previously compared to after the complications are there. So after this, I just want to share this cartoon that scientific discoveries are exciting, definitely, but they carry with them a responsibility to use the knowledge with wisdom. So you should ask ethical questions. You should have people whom you can turn to understand the limitations, and then you have to explain it to the parents accordingly. Thank you so much. And I would like to thank all the clinical collaborators who shared the patient data, and they involved MedGenome for carrier screening. And I thank my entire team. And again, I thank Dr. Mandakini and Dr. Priya for involving me in this talk today. Thank you so much.
Okay. Thank you very much, Dr. Sheetal. I think we will move on to our next speaker. So our next speaker is again our own Dr. Priya Kadam. She is the Associate Director for Reproductive Genomics at MedGenome. Dr. Priya completed her MBBS from Bangalore Medical College, and she has a Master in Science by Research from National University, Singapore. She has filed for patents in the US and China for her work on prenatal testing. Her work has been published in journals such as Journal of Proteomics, Cancer Biology, Therapy, and among others. And she has been associated with MedGenome for the past 6 years and oversees the prenatal testing at MedGenome. So over to you, Dr. Priya.
Thank you, Joseph. I'll just share my screen right now. Okay.
Yep, you're good to go.
Yeah, okay. Good afternoon, everyone. Thank you for joining in this webinar. And I'd like to begin by thanking Dr. Pradhan for really involving us in this series. We have enjoyed listening to various lectures, and today we had 2 excellent presentations. So this is a very good medium for us to connect with the audience and also tell you of what the tests that are available and where you can use it in your own practice. Okay, so I will be talking about non-invasive prenatal testing, and most of you might be aware of this test, but I will actually talk at some of the aspects which we see from this side in the laboratory, and also some of the new applications which are not really fully developed yet, but something which are very exciting and which can be used and will come in the future. So like I said, so a little bit about the background of NIPT. Most of you as obstetricians may be aware, might have offered the test to your patients, but like I said, I'll go into some more detail. And the key discussion points, the questions that we get frequently, what amount of fetal fraction is enough? Is it safe in, I mean, is the sensitivity the same in twin pregnancies? Should we use it to screen for microdeletions? What about rare autosomal trisomies, single gene disorders, and some of the limitations which you should be aware of? Though they are pretty few, it's good to understand the scientific reasons behind it. So, I mean, of course, we have been in COVID times, and we did see a kind of a jump last year for NIPT, because one of the major fallout, not really fallouts, good effects actually of NIPT is that it reduces the number of invasive testings for certain in certain situations. So that kind of reduces your exposure in hospitals and such things. So it has been useful, we see, in these conditions as well. Now, as obstetricians, all of you are aware why chromosomal screening is important, because you know, all women would desire for a normal baby. Most of these conditions occur spontaneously. There is also increase in the number of women giving birth later on in life, and that increases the risk of chromosomal abnormalities, as all of you know. And also, these are relatively common. Dr. Shagun has spoken about single gene disorders, which is about 1 in 100 taken together. But if you look at all abnormalities together, it would be about 1 to 2%. So this is something which is not so uncommon, and therefore screening of all pregnancies is important. This is pulled out from Dr. Verma's paper, which shows chromosomal disorders. If you're looking at to begin with for NIPT, it is about 1 in 166. So you would expect about 1.5 lakh babies to be born with disorders if no prenatal screening is done. Now, when you want to look at an obstetric population, there would be what are the types of tests that you can offer? There will be screening tests and there'll be diagnostic tests. So screening tests are those in which you would offer to a large group of people, ideally to every pregnant woman, and then identify women who need to be evaluated further. And these women, of course, you would do a diagnostic test and based on counseling, go further. So what are the screening tests which are currently available? What we are talking about now is the cell-free DNA, the first trimester screening, portable screening, and NT and the ultrasound alone, which all of you will be aware of. Now, why this test has taken off so much is because it can be offered from 9 to 10 weeks of pregnancy, and as such, you can test anytime during pregnancy. There is no time limitation. Then the detection rate for trisomy 21 is the highest, as you can see, over 99%, and the false positive rate, which is actually not mentioned here, is less than 0.1%. So as opposed to all other tests where it is higher. And the other thing which I'll talk about is the positive predictive value of NIPT compared to the other tests. Now, what are the diagnostic tests? As you can see on the right side of the screen, the diagnostic tests actually would be FISH or karyotype or a chromosomal microarray. And the procedures that you do, of course, will be chorionic villus sampling or amniocentesis, the most common ones. But one thing we would like to stress is that whenever you do NIPT, we would recommend a background ultrasound to be done, because we need to know the number of fetuses, whether there is any gross abnormality, in which case NIPT will not be the appropriate option for testing. So what is non-invasive prenatal testing? And in the previous 2 lectures, actually, we have seen the types of chromosomal disorders, type of genetic disorders which are present in humans. So you would have a aneuploidy, or you would have a microdeletion, or you could have a single gene disorder. Now, NIPT actually has the potential to detect all of these disorders, these disorders from the maternal blood during pregnancy. But right now, it is well accepted for trisomy 21, 13, 18, and sex chromosome abnormalities. And it's called non-invasive prenatal testing, but you still need peripheral blood of the mother. And it is compared with invasive testing, but we must always remember that it is a screening test, though it is highly accurate. Now, it is known that what is the material actually we use for testing? It's known for more than 100 years that material from the fetus, the genetic material from the fetus actually goes to the mother, and also mother's material goes to the fetus. Now, this can be in the form of a fetal cell or fetal DNA, RNA, or protein. What we actually look for is this cell-free DNA, that is the DNA which is outside the cell. So every individual will have some amount of cell-free DNA in their blood. And when a woman is pregnant, the cell-free DNA from the placenta actually will circulate in her blood. And it is this cell-free DNA that we use for testing of any of these abnormalities. There are some of very important facts about cell-free DNA which all of us should be aware of, because it has implications on your interpretation of the test. We know that it was discovered in 1997, and your cell-free DNA actually has the mother's component, mother's cell-free fragments, as well as the baby's. Okay, so this is very important to remember. And your fetal fraction actually is about the fetal component of the whole cell-free DNA. So which is about 5 to 25%. And also that it comes from the placenta. So this is very important to remember, because sometimes there can be discrepancies between the result that you get from the cell-free DNA and the placenta versus the fetus. More than 99% of the time it is going to be identical, but sometimes there can be differences, and which we will speak about a little later. And another thing is that the cell-free DNA from the fetus actually clears rapidly after the baby is born. Within 24 hours, it's completely gone. Therefore, the test that you do during pregnancy will pertain to the fetus, current fetus. Now, when cell-free DNA testing first came in about 2011, there was a lot of excitement, and many professional societies outside came up with guidelines. And since it was very new and optimized in high-risk individuals, they decided that it is best to do it in high-risk patients only. But slowly and gradually, as the efficacy of the test and more evidence became available, the test was then expanded to be used in a lot of other situations, including average risk and general risk populations. ECOG guideline recently in 2020 has actually recommended it, or the fact that it has to be offered to all patients which come for antenatal tests. And actually in 2016, again, the ACMG guidelines have said that the non-invasive prenatal testing can actually replace conventional screening for Patau, Edward, and Down syndrome across all maternal age spectrums and from 9 to 10 weeks of pregnancy. Now, what has been the impact since 2011 that this test came in? You can see this blue graph of the test being adopted, and this red line actually shows the reduction in the number of invasive procedures for this purpose. As we know, NIPT actually now is spread worldwide and it's available almost everywhere in the world. We were discussing that once large-scale validation in 2011 became available, the test became commercially available in 2011. At MedGenome, we finished the validation and started offering the test from 2015. So we have done for over 5 years, we have been offering the test, and we have done 2 different platforms: the SNP-based testing as well as the counting-based technique. Now, what are the most common indications? So in your practice, when do you actually offer NIPT? The most common, of course, is actually it is the abnormal serum screening. So mostly in everywhere else I see in India, the samples that we get is when there is a high risk on maternal serum screening. That is the most common. Advanced maternal age on its own sometimes. If there is some ultrasound findings which are suggestive of aneuploidy, and if there is a family history of chromosomal abnormalities. Looking at our data, we see about 40% of the cases come because of the high risk on serum screening. Now, what are the major advantages on why NIPT is so widely used? One is because of high detection rate, which we spoke about, more than 99% detection rate, lower false positive rate compared to other screening methods, reduction in the number of invasive procedures, and the high negative predictive value. That means usually when you have a low risk on NIPT, the risk of having this, the abnormalities which you have tested for becomes very, very low, though not completely zero. And risk assessment is independent of age of the patient. Now, once this test came in, of course, it had to be compared to the standard screening which was available then. This is a paper from 2015 which actually looked at the performance of standard screening versus cell-free DNA. This is the data for trisomy 21. As you can see, the sensitivity for standard screening is 78% versus 100% for cell-free DNA, and specificity is 94 and 99. But what is most striking is this positive predictive value. So you see the positive predictive value with a cell-free DNA is 80.9%, while for standard screening it is 3.4%. So that is a huge difference. And when you come to trisomy 18 also, it is a very high. You would have 14% on standard screening, while 90% on cell-free DNA. For 13 it is 3.4 versus 50. So what does this actually mean? Okay, so if you have 100 cases, okay, which have tested high risk on first trimester screening, and 100 cases which has tested high risk on NIPT, most of those cases on NIPT will actually be true positives. That means it is a highly accurate test. Okay, so that is the positive predictive value, 91 over here, and 3 on the first trimester screening. So, this is one concept which is why it becomes so much more useful for the clinicians. Now, this is one case which I like to put in here, because this was a case in which the regular screening was done, but it came as low risk. But the testing that was done was triple marker screening, which I know now is a bit is kind of obsolete, and the baby was born with Down syndrome. So you see, this falls in the false negative category for that particular test. So the false negative rate with NIPT, though it exists, it is much lower than your standard screening. Now, when we started offering the test, actually, we wanted to see the efficacy of the test in the Indian population. So this was a result of the study which we did with Sir Gangaram Hospital and many other about 10 reputed institutes, where we looked at about 500 cases, and sure enough, the sensitivity and specificity was comparable with international data, and the positive predictive value also was comparable. And this was about 500 samples in which we had complete follow-up till the baby was born, or in high-risk cases where invasive test was done and the karyotype result was available. So in addition to that paper, there was one more interesting case which we were able to publish, and this was the ability of NIPT to detect a variant of Turner syndrome, which when they did an invasive test, the fish was abnormal. Fish was normal, but the karyotype further showed a abnormal Turner syndrome with an isochromosome X. So like a typical case scenario of where we get or NIPT will be useful is this particular case, where this was this 36-year-old woman, advanced maternal age. On combined screening, her risk was pretty high, which was about 1 in 107. She might have gone in for invasive testing, but she was offered NIPT, and that turned out to be low risk. So with the negative predictive value, with a high negative predictive value, she was appropriately counseled, and on genetic counseling, she continued with the pregnancy. A normally scan was normal, uneventful pregnancy, and a normal full-term delivery. So now very often we get questions about fetal fraction. Like I said before, fetal fraction is the amount of cell-free DNA from the fetus in the whole cell-free DNA. Okay, now what is the cutoff? So a few years ago, the cutoff was about 4%. Okay, so but now with other technologies and with advancing technology, actually the cutoff is anywhere between 2 to 4%. But what is important is the validation. And one thing to remember is, if the fetal fraction is lower, your sensitivity is slightly lower compared to a higher fetal fraction. With higher fetal fraction, you are much more confident of the result. And another thing to remember is, if you get repeatedly low fetal fraction and that the laboratory does not give you a result, there could be a small possibility, which is about 3 to 5% possibility of an aneuploidy. So that could be trisomy 18, or it could be trisomy 13, or it could be triploidy. And most of the international guidelines recommend that you always look at the fetal fraction. Now, two important points with fetal fraction is that the fetal fraction decreases with maternal weight, increasing maternal weight, but it should not discourage you from offering NIPT to individuals who are overweight or obese. And that's because most of the cases you will get a result, but some amount of genetic counseling to prepare them that there might be a need of a redraw is something that helps in patients' compliance. And about 70% of the cases, wherever there is a redraw, we do get a result. The other thing is with increasing gestation age, you would expect the fetal fraction to increase. So if the test is done very early in pregnancy, sometimes you might get a low fetal fraction result. So one other example I'd like to share, there was this primary gravid actually, she was 39 years old and her risk on combined screening was 1 in 7 to 9. So this falls in the intermediate risk category. So many times we get cases where the risk falls in the intermediate risk on maternal serum screening, but this actually tested as high risk for trisomy 21. She was appropriately counseled to go in for confirmatory diagnostic testing, and on follow-up, I mean, she did the testing, and as you can see the FISH report here, there are 3 signals for chromosome 21, and the karyotype also showed 3 chromosome 21 in her result. So she was counseled further and she decided to terminate the pregnancy. There are a couple of other interesting cases which I'd like to share. There is this lady who came, she was 32 years old. This was her first pregnancy. It was a spontaneous conception. And we gave her a result of monosomy X. The fetal fraction was very good, 10.5%. Now, sometimes what we have observed is despite counseling, the patients go in for termination without confirmation, which is actually not to be encouraged, because sometimes we will go later on and figure out that there can be a possibility of a false positive on an NIPT, and therefore a confirmation is absolutely essential. But still, maybe if there are other findings or some other social reasons, we have seen terminations without confirmation of a NIPT result. Okay, so for this case, they actually terminated without follow-up. But they wanted to actually know what was the status of the fetus. They sent the products of conception for a chromosomal microarray, and sure enough, actually it was a Turner syndrome, but it was a mosaic Turner. That means about 30% of the cells were abnormal. Now, now this actually kind of strengthened their faith in NIPT. And next time when she was pregnant the following year, she came back and was worried about the previous history of monosomy X, and the NIPT was normal. Now the pregnancy is continuing well. This was another very interesting case in which we had a pregnant woman herself with Down syndrome. So initial counseling that we did to the clinician, we said actually it is not recommended because we know that the background will be full of trisomy 21. So it will not be possible to actually identify the fetal result in this. And sure enough, but I mean, the patient was not willing to go in for an invasive testing, but so they sent the NIPT sample, and the NIPT tested high risk for trisomy 21. And at this point, we said, no, you really need to confirm in the fetus, because we know the background will be trisomy 21. This will be a problem with the counting method. And they were able to convince the patient to go in for invasive testing, and the fetus actually turned out to be normal, as you can see here. Okay, now that is about the common chromosomal abnormalities, that is trisomy 21, 13, 18 and sex chromosome abnormalities. Now what about expanding it to a general population, microdeletions, copy number variations, twins and rare autosomal trisomies? Now when you expand the testing, there are several considerations. The first is, what is the clinical relevance, okay, of actually offering this test for these disorders? Is there validation data data available and what are the detection rates. So these are the common things which you would be thinking about. And the concerns again will be higher false positive rates, and also if you find an abnormality, you might have to go in for an invasive testing, and the number of invasive testings may increase, and the counseling becomes extremely complex. So initially, like I said, the test was offered to high-risk individuals, but now there is enough data to say that it will work just as well in a low-risk and an average-risk population as well. But the positive predictive value will be slightly lower in the general obstetric population as opposed to high-risk population, because it is also a function of prevalence of the disorder. Now, but whatever said and done, as opposed to a standard screening, NIPT will definitely give you, standard screening would give you a positive predictive value of 3.4%, and NIPT on all populations taken together would still give you about 80% positive predictive value, and for average risk population, slightly lower. Now, should you use NIPT test for microdeletions? Now, there are certain common microdeletions for which NIPT can be offered. We have experience for doing it for about 5 microdeletions. The previous speakers, Dr. Shagun, has already described what microdeletions are. The most common microdeletion actually is 22q11.2 microdeletion. So there is a small portion of chromosome 22 which is lost, and it is associated with a typical syndrome. Now, with the availability of technology, you can see there is an explosion of identification of these disorders. So there will be a group of disorders which will actually fit into a syndrome, and more of these disorders have been identified with advancing technology, especially chromosomal microarray. Now, we have experience with dealing with these 5 abnormalities, that is 22q, 1p, 36 deletion, Angelman, Prader-Willi, and Cre-Duchamp. Now, what could be spoken in favor of it is that, see, in most of it, you will not have any antenatal finding. And so far, there was no test to actually look for these disorders. And therefore, it would have been missed. And many of these disorders are actually extremely serious, as in they could cause severe intellectual disability. And as we know, there are no cures. Now, if you look for at 22q11.2 deletion itself, the risk is, the incidence is actually higher than that for trisomy 18 and trisomy 13. Okay, so it does make sense to look for 22q11.2 because an overall incidence can be anywhere from 1 in 2000 to 1 in 4000. And the risk of these disorders actually remains the same, about 1 in 1000 throughout the reproductive age of a woman. That means in younger women, you would expect these disorders to be more common than Down syndrome. So there have been various data which shows a high detection rate, but the positive predictive value as such is quite low as of now. And both targeted and genome-wide methods actually can help look for these microdeletion syndromes. The positive predictive value, as you can see, is about 3.8 to 17%. We do have a little bit of data for microdeletions. Only about 8.2% of all NIPT samples opt for microdeletion tests, and we've had 5 positives or high risk so far, and one of them actually is a true positive. But if we look at guidelines, as such, we don't have guidelines for this in India right now, but for what ACMG or any other guidelines, they do not recommend genome-wide CNV so far. So those were microdeletions. Now, there are also variations which may be of a slightly bigger size. Now, the genome-wide technology can actually look for copy number variations which may be slightly more than the microdeletion. So that may be greater than 7 MB. So these deletions can actually be identified through NIPT. Okay, now we know what is the need for testing. We know that about 1.7% of general population will have clinically relevant subchromosomal CNVs. Okay, and about 30% of them may be more than 7 MB. So therefore, it makes some amount of sense to be screening for these copy number variations. So proof of principle studies have been around for about 5 to 6 years. For CNV detection, the detection rate is about 74 to 88%. And one of the advantages would be it would give an early clue to potential abnormality before ultrasound abnormalities appear. And the other thing which has been noticed in literature is that many of these are associated with abnormal first trimester screening. So that itself can give a clue that there may be a CNV present in the fetus. The difficulties, of course, is if it is a variation which is not very well known, then there could be a genotype-phenotype correlation issues that can be very tricky, and counseling becomes very difficult. And like Dr. Sheetal did mention the gray zone of variant of unknown significance, then a counseling can become very difficult if we look for genome-wide screening and then we try to transfer the knowledge to the patients. Okay, and so far none of the professional societies actually recommend this, and the sensitivity depends on the fetal fraction, the size of coverage, and the depth. But having said that, let me just bring to your attention one particular very interesting case that we had. This was in this was a case which came last year. It was a 13-week pregnancy, a 29-year-old woman, but her risk was pretty high, actually 1 in 36. She did not want to go for invasive testing. She went in for an NIPT. For the conditions that we tested, 21, 18, 13, it was a low risk. Okay, but around 37 weeks, the clinician called back and asked us, I know that you all do genome-wide NIPT. Is there something else that you have found, any other CNV or anything? Because they are seeing some abnormalities in the fetus, and this was much later. They observed this much later. There was a severe IUGR actually. There was abnormality at the tip of the spine, sacral agenesis, and she asked to look for the data. And actually in our data, we do not report this as of now because there is no validation, but we did see that there were actually 2 copy number variations in chromosome 6 and chromosome 7 of sizes about 12.5 and 10.8. But at 37 weeks, they decided the best way to approach this was to wait for the birth of the baby. The baby was born, and on a chromosomal microarray, both these abnormalities were confirmed. So this is just a proof of principle that it is a kind of useful test to be able to detect copy number variations much early in pregnancy, and so that they can be confirmed and then and appropriately dealt with. Okay, now what is the efficacy in twin pregnancies? Now we all know that incidence of twin is increasing. It was increasing, and now some of the graphs show that it is decreasing again. We know that it is associated with higher risk of complications, and if you're trying to do a serum screening for chromosomal abnormalities, it is difficult because sensitivity is low and false positive rate is pretty high. And if you want to do an invasive test also, the risk of complications are more, and also of like the risk of complications are more. And also one thing to remember is that to know the zygosity, chorionicity is very important, because the information of whether it is monochorionic or dichorionic is important for us, because we need to understand whether the twin, it is likely to be a monozygotic pregnancy or dizygotic pregnancy. So we frequently get this question whether the sensitivity in twin pregnancy is the same as singleton. So there was this meta-analysis in 2019 where Dr. Gill actually looked at a lot of data. And after that, there was also a guideline from ISPD. They have actually combined Gill's data as well as other data, and they have found this is actually the sensitivity which you can look at. It is, and this is across platforms. Okay, it could be the SNP-based platform, or it could be the counting method. So you have about 98.8% for trisomy 21, 93% for trisomy 18, and 75% for trisomy 13. So these are the figures which are like a combined or meta-analysis score. And initially, actually from 2015 onwards, most of the organizations were silent on whether it should be offered in twins, and triplets, actually they are still silent. But now more and more more and more societies are actually recommending that twins, NIPT in twins actually has a sensitivity pretty much similar to that of singleton pregnancy. So this was the latest guideline which came in last year in 2020. So it says first trimester cell-free DNA screening for common autosomal trisomies is appropriate for twin pregnancy due to sufficient evidence showing high detection rate and low false positive rate. And of course, you have to confirm it by invasive testing. Now, a little bit of our data, we have, as expected, it is about 2% of all cases, and about 2/3 of them are artificial reproductive techniques, and have been used. And one thing to remember is there is a slightly increased chance of insufficient fetal DNA, because for twin pregnancy, we actually require double the minimum amount of DNA to be present for us to give a confident call. And so far we've had 3 high risk, and one of them has been confirmed, and two trisomy 21s have been confirmed, and the trisomy 13, there was no confirmation. So one thing to remember about twin pregnancy is that the risk is for the whole pregnancy. We cannot tell which individual fetus is affected, and it is the high risk result requires confirmation of both the twins. That means an invasive testing has to be done for both the fetuses. And but if you get a low risk or low risk, it has a very high negative predictive value. Therefore, you can rest assured. And of course, it requires a slightly higher fetal fraction. And higher than twin pregnancy, that is triplets and quadruples, cannot be tested as of now. Now, the other, I'll go very fast now because I see that we've gone much beyond. Okay, so rare autosomal trisomies. So what are rare autosomal trisomies? So these are trisomies other than 21, 13, 18 and sex chromosome abnormalities. Now NIPT, genome-wide NIPT, we look at all chromosomes. So sometimes we pick up these abnormalities. So the incidence of these rare autosomal abnormalities is about 0.3 to 0.4%. And the most common are trisomy 7, trisomy 16, and trisomy 22, which are actually similar to CVS data. And this again confirms that your cell-free DNA is coming from the placenta. The POC pattern is a little different. Okay. Now, is it important to screen for these, or do you need to have this in your report? Now, we know that, there hasn't been too many studies for this, but about 10 to 15 studies actually say that 50% of this would have a normal outcome, but other 50% will not have a normal outcome. So they have a risk of, adverse obstetric outcomes like miscarriage or malformations. There could be intrauterine growth or postnatal growth retardations, and also there could be one other problem. So what are the mechanisms? Okay, so now you see, if you have a rare autosomal trisomy, one thing, in 80% of the time, it will be confined to the placenta. Okay, so therefore, if you screen the fetus, then you have to do an invasive test of the fetus. The fetus will be karyotypically normal. So that is one of the most common mechanism of detecting this rare autosomal trisomies occurring and the fetus developing normally. Okay, so in some of the cases there may be a true fetal mosaic, that the fetus also may be a mosaic, may be carrying a mosaic, and that's why it can get to that stage of pregnancy. There may be a uniparental disomy. Sometimes it may be a vanished twin, which might account for that result of a rare autosomal trisomy. There could be abnormalities in the mother as well. Okay, so the mother may be a mosaic, or the mother may have malignancy. So which might show up as a very bizarre result on an NIPT. And one second, this I will just close. Yeah, and some of the cases may be unresolved. Now, yeah, like I said, what the figure here shows, 50% of the cases will result in live births. Now, what are the benefits? Benefits of this expanded screening is it might explain some of the adverse obstetric outcomes, and also it helps in identifying high-risk pregnancies, because they are at a risk of IUGR, they might be at a risk of sudden intrauterine death. But of course, the cons again are you might have to do an additional invasive test, but I don't see that as so much of a problem, because the incidence itself is about 0.4%. So it is not going to increase much more than what you would normally do for a common autosomal trisomy high risk. Okay, and there are many of the societies, actually this is a hot topic for debate, and it is found that people are kind of totally balanced, like 50% before and after the debate. We do have some experience with this. We have about 4 cases which were about trisomy 16, trisomy 9, trisomy 7, and on testing the fetus, the karyotype is normal. But having said that, it's important to remember that if there is an abnormality in the placenta, it could cause some fetal complications. So this again becomes a high-risk pregnancy. Now let's come to the last but one topic, single gene disorders. Okay, can NIPT be used for single gene disorders? As you can see in this cartoon, see if there is you have a couple, this is the mother, this is the father, which has an abnormal allele over here. So the fetus formed is like this, or the fetus formed is like this. So if the fetus gets the abnormality from the father, and it is present only in the father, you will be able to detect it like this. Okay, so this is for paternally inherited disorders. So this looks pretty straightforward. So the common examples of this for paternally inherited or de novo, that means they are not present in the father but can be seen, might occur spontaneously. Those are de novo disorders. So those can these tests actually has been offered for achondroplasia, Epert syndrome, thanatophoric dysplasia, and the approach is actually to exclude or detect the presence of paternally inherited or a de novo mutation. So the most common approaches would be a PCR or a digital PCR or NGS. Now, these are for autosomal dominant disorders. What about autosomal recessive? And in situations where both parents carry different variants. In that case, if you can exclude the paternal allele, okay, then it reduces the risk, because now for autosomal dominant conditions, you need to have both the alleles which are defective. You will be able to exclude the paternal allele, or if you detect the paternal allele, that means there is a 50% chance that the fetus is affected. So you cannot actually exclude the maternal allele being affected, because the background will be there of maternal cell-free DNA. So in this case, you would have to do an invasive testing. And of course, the common method would be PCR, digital PCR, and NGS. This is for common autosomal recessive disorders like cystic fibrosis, thalassemia, congenital adrenal hyperplasia. And what happens if it is an autosomal recessive disorder and both the parents are carrying the same variant? This is the trickiest thing, as you can extrapolate. Okay, so in this case, we need to estimate the allelic ratios, and there is a need for accurate detection of fetal fraction. So for the situations in which this is tried in is sickle cell anemia. So there are some companies which actually offer these tests, but they haven't gained much more acceptance as of now. The reason maybe it is there isn't nod from professional societies yet. There can be difficulties when the fetal fraction estimate has to be accurate, and also there could be unexpected maternal complications, or there may be placental mosaicism. Okay, but they have been offered for advanced paternal age, or if there is an absence of family history. So like Dr. Sheetal said, Dr. Shagun said, if you look at all single gene disorders, okay, that the risk is about 1%. Okay, so does it make sense to actually test for more and more of these conditions in the prenatal stage non-invasively? The question is still open. Of course, for this, we need to get a lot more data and lot more validation. Okay, and like I said, the guidelines still do not recommend it, stating that there isn't enough data. Now, what are the current limitations and challenges? Let me just go over quickly. With NIPT, as with any other screening test, you would expect false positives, false negatives, or there may be test failure, or sometimes you might have incidental findings like maternal neoplasia. I did mention that before. Now, this cartoon, actually, I like to talk about this because it clearly explains how a false positive or a false negative can happen. So this is when the fetus is developing. This is, the embryo, zygote actually, there will be a 2-cell stage, and one of the cell has become aneuploid. So this is trisomy. And then if it continues dividing, dividing like this, you will see that sometimes it may be present only in the inner cell mass, sometimes it may be present only in the trophoblast layer, here and here. And here it is present in the inner cell mass as well as in the trophoblast layer. So now in this situation, the situation B, you would see that this abnormality will be picked up because it is present in the placenta. And when you do an invasive test and test the fetus will also be affected. So this will be a true positive. Now, if the abnormality is present only in the placenta, so this is actually seen with the common autosomal trisomies also, and also with rare autosomal trisomies, which we just discussed. Okay, so you will see a high risk on NIPT, but if you test the fetus on karyotype, it will be normal. So this becomes a false positive, and this is also a similar scenario in which it becomes a false positive. But this is the most dangerous scenario in which the trophoblast cells are normal. So the cell-free DNA which you use for NIPT testing will be normal, but the fetus will be affected. Okay, that's because the fetal cells are abnormal. So this is a false negative scenario. I just like to bring this in because you need to know the possibilities of how these things arise. The very last thing, I'll just touch upon vanishing twin pregnancy, because very frequently we get this question, should NIPT be used in vanishing twin pregnancy? We'll just go over this one case. There was this 37-year-old woman who had a vanished twin at 6 weeks. We didn't know of that history. Actually, this came much later. When we did the NIPT, we got this result of vanishing twin pregnancy or a possible triploid. So this is only the SNP-based testing or the panorama testing which can actually give you this type of result, because it actually identifies the number of genotypes. So when we got this result, I mean, as such, NIPT is not recommended in vanishing twin pregnancy if you're doing it by counting method or any other method, because you will not get a result for the existing fetus. Okay. So in this case, again, invasive testing was not done, but the repeat sample was sent at 24 weeks of pregnancy. And in that, at that time, we were able to give a low risk result. So the fetal fraction was about 17%. That means, I mean, what we can assume here is that the DNA from the vanished twin had vanished. Now, again, people ask, how long should we wait before we send the sample after the fetal demise? That, again, is a very tricky question, because we have seen sometimes it can be as low as 5 weeks, and sometimes more than 12 weeks also, the DNA may still be present. There is this particular paper which has says that, which has actually systematically studied that and said that up to 8 weeks, you might still be able to see the fetal DNA. Okay, so therefore, as such, vanishing twin pregnancy, if there is a history, NIPT can be confounding. Okay, so you have to be extremely careful. There is a high possibility of a false positive because the vanished twin might have died or because of an abnormality. The guidelines discourage it, and there is no way of knowing. If you send a sample and there is no history, there is no way of knowing whether the DNA from the vanished twin is still present. Okay. As Dr. Sheetal also mentioned this quite a bit about pre-test and post-test counseling. As with any other genetic test, this counseling is very, very important because it sets expectations, and also they should kind of understand. I mean, I've put in a lot of points over here, but what is important is the patient should know that if there is a high risk, it should go in for further confirmation. And if there is a low risk, it means it is a low risk for the conditions which are tested, not everything else. Okay, just for the conditions which are tested. And there is a possibility, a rare possibility, that the fetus may have some other abnormality. Now, post-test counseling also, once the result comes in, also it is very important to kind of explain the result to the patient. So I'd like to conclude here by saying NIPT is a very sensitive test. Now, with common chromosomal abnormalities, there is also a potential for expansion into copy number variations, into microdeletions, into single gene disorders. And the greatest advantage that we can see is that it significantly reduces the need for amniocentesis, need for invasive tests and the associated risk. And because of the high negative predictive value, it gives reassurance. chromosomes. And of course, the jury is still out whether it is still relevant to do it for microdeletions and for rare autosomal trisomies and single gene disorders. And like with any other genetic test, both pre and post-test counseling is very important. And of course, we must look at it as one of the tests in whole antenatal care. So you must correlate it with the ultrasound findings. Yeah, these are the people from the lab, and I'd like to thank you all for your attention. Joseph?
Yeah, thank you very much, Dr. Priya.
Dr. Mandakini.
Yeah, please go ahead.
Excellent. Every slide, every bit of it was full of case discussion, case scenario that we come across day to day, and all three talks: single gene disorder, screening for genetic disorders, and NIPT and extended NIPT, touching upon almost every clinical aspect. So that is the reason why more than 100 people after 2 hours is still connected. Usually they leave. So these are true positivity. That means truly they are interested to learn genetics. So these are the people who are still connected. And I must congratulate all of them for having this wonderful session. And looking forward to have more sessions like this. Because we have exceeded time, so Joseph, can we call off and then take up all those questions in their personal email?
Yes, ma'am, we can do. One or two questions we can address.
Okay. Okay. Yeah.
Yeah, there's one question. Some lab reports NIPT for all 23 chromosomes. What is the reliability of such reports?
I think the best thing is actually go back to them. Sometimes in the report itself, they will give you a table. If they are not giving you a table of what is the sensitivity, you might have to ask them, because it's very important to know the validity. The reason why we don't offer all chromosomes is because we need to do a validation. Right? So we are actually in the process of doing the validation. And the other point to remember is these chromosomes, I mean, these aneuploidies are very rare. Okay, so a validation as such is difficult, but it is very important to look at the clinical aspect of it, whether there is any abnormality in the fetus, and then see whether, sorry, one thing to remember is once you have an abnormality in any of the other chromosomes, it is still important to do a testing in the fetus. You need to do a confirmation in the fetus. So I think you have to go back to the lab and ask them what is their validation. Sometimes it is there in the report itself.
Yes, I do agree. That first thing is see your fetus, again, do an ultrasound. Somebody has asked, if I'm doing an ultrasound, if I'm doing a cytogenetic test, why should I do an ultrasound? Or if I'm doing an ultrasound, then why should I do a cytogenetic test? And probably Dr. Seetal wanted to answer that.
I'm trying to type for a long time, but I think I will answer it live.
Yeah.
Because everybody knows that all malformations may not be due to a specific genetic disorder. So, all genetic disorders may not be detected by what the tests we do. So there are limited number of tests we are doing. So doing a genetic test doesn't exclude that the child will be born without any malformations. And malformations can occur right from conception to end of pregnancy also. So certain skeletal dysplasias may come later on, but you don't screen for skeletal dysplasia in a routine test. So even if you are doing whatever genetic test, there is always a recommendation that the ultrasound follow-up has to be done as per the recommendations of the Gynecology Obstetrician Society.
Yeah, I do agree with this, fully agree. Ultrasound cannot replace cytogenetic, cytogenetic cannot replace ultrasound. And yesterday only I received a photograph of serinomelia, that means fused legs, where usually it is not genetic. So you will, even if you do exome sequencing, you will not find a genetic abnormality for it. So it is not mutually exclusive. Somebody has asked that CF DNA is self-fetal DNA is cleared after 24 hours of delivery. Why not it doesn't disappear in vanishing twin? That's an excellent question. Why it doesn't disappear in vanishing twin?
What I think is because the gestational sac actually is still there, so the remnants of the fetus are still there, and the reabsorption process is very slow. But once a delivery happens, the placenta is also delivered, right? So the placenta is actually out of the uterus, and
So
that's why it disappears. Yeah,
it's most likely the explanation. Vanishing twin at about 6 weeks. How to know if doing ultrasound at 12 weeks for first time ever? You cannot know. If it has disappeared, you can't know. Anything else, Joseph?
Nothing else, ma'am. I think we have covered most of the questions.
NIPT can be offered as early as 9 to 10 weeks. What is the basis on which we can offer it? Indication or which patients to offer at that early gestation?
Yeah. May I answer? Yeah.
Yes.
Technically, we can do it from 9 to 10 weeks of pregnancy, but ideally you should do it after your NT scan. That is an ideal thing, because what we have seen in terms of fetal fraction, and also we need to know how many fetuses are there, whether there are any gross abnormalities, in which case you might not do an NIPT at all. Right?
Yeah, true.
That is the reason. Yeah.
So true. So this was an excellent session. Again, I thank all the speakers and our partner MedGenome for sponsoring this event, and from FOGSI and the society, Genetics and Fetal Medicine Society. I thank you all. Thank all the participants for joining it. And if anybody has got any query, they are free to ask in the email provided, or the society has been asked to send it in my email. The committee will look into it. Thank you all, and we will be Joseph, you will be sending the certificate. I'm
Yes,
sorry that I have not been able to send the certificate of previous participation also, because I myself have not finalized, and it has to be approved from FOGSI. And today's participation also, we will do the same. Joseph,
sure.
Anything else, or we conclude?
No, ma'am. So yeah, on behalf of MedGenome, I'd like to thank Dr. Mandakini for providing us this opportunity to present to this audience, and the excellent talks that we had over the 3 sessions. I'm sure we've concluded this series, but we'll be back with more series in the near future. And we look forward to the audience participation. And also thanks to all the speakers who've been part of the series from the beginning, and today's speakers also, Dr. Shagun, Dr. Sheetal, and Dr. Priya, for taking time off and presenting. And the audience, like you mentioned, have been very consistent. They've stayed despite, going over the time. It shows their commitment and their interest to learn, and we really appreciate that. And so the audience, as ma'am said, we will be sending out the certificates as soon as it's approved. And the video also will be sent out in a week or so for the recording, so that you can go back and visit that. And if you have any additional questions, you can reach us at the toll-free number below, and at diagnostics@medgenome.com. And hopefully we'll be able to answer all your questions. We have more webinars coming up. We'll keep you informed on that through your email. So please look out for them in your email. So so that's about it. So thank you very much, everyone, and thank you for the opportunity.
Thank you. Thank you, Joseph. Thank you, Joseph, for organizing.
Thank you very much, Dr.
Yeah. Thank you. Thank you.
Thank you.
Bye-bye.
Bye-bye.